Hey everyone, thanks for joining me today for episode 771 of People Behind the Science. I'm your host, Dr. Marie McNeely, and I'm joined today by our guest, Dr. Samantha Maragh, to talk about life and science. This episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And today, our guest Samantha is going to tell us more about her lab and her life. So get ready to meet another one of our fantastic people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Samantha Maragh. So, Samantha, welcome to our show today. How are you?
I'm wonderful. Thank you so much for having me.
Well, Samantha, I am so happy that you're here with us today, and I'm excited to learn more about you and your work. Listeners, if you haven't met Samantha yet, she's leader of the Genome Editing Program and co-leader of the Biomarker and Genomic Sciences Group at the U.S. National Institute of Standards and Technology, or NIST, NIST for short. She also represents the U.S. as a technical expert on nucleic acid measurements for the International Standards Organization, or ISO, Technical Committee on Biotechnology. Samantha received her bachelor's degree in biology with a specialization in cellular and molecular biology and a minor in chemistry from Loyola University. She went on to get her master's degree in biotechnology, molecular targets, and drug discovery from Johns Hopkins University, and her Ph.D. in human genetics and molecular biology from Johns Hopkins School of Medicine. In 2019, Samantha received the Outstanding Young Scientist Award from the state of Maryland, the Maryland Academy of Sciences, and the Maryland Science Center. She was also selected as the recipient of the George A. Uriano Award in 2021 for her success in building the NIST Genome Editing Consortium as a public-private partnership. And in 2022, Samantha received the Measurement Science Excellence Award from the NIST Material Measurement Laboratory for leading the development of the first international standard for the field of genome editing published in 2021 and deploying the first interlaboratory study for the genome editing field. And Samantha, we are looking forward to learning more about you and your work today, but perhaps we can start by learning more about you. So can you tell us, what do you like to do when you're not doing science?
So when I'm not working, I both love to eat and make food. Probably not so strange to people, but I find a lot of freedom in the creativity of making food. Sort of like science that I can be a little bit more flexible with. I can go a little bit rogue. Don't have to follow my protocol exactly like in the lab. I also love to sing, and I sing regularly, particularly on Sundays at my church. And I love little games that turn my brain off after lots of critical thinking in the lab, just kind of mindless puzzle kinds of games.
Well, it's wonderful to hear about some of these different facets of your life. You mentioned you enjoy cooking and making food. What are some of your favorite things to make?
Oh, I really love seafood. And so I try to make the things that I really love. So if I go out and I find something that I like, I try to make it, as long as the magic behind getting it out isn't, because those ingredients are really hard to source. I veer towards seafood and other kinds of meats. I'm a bit of a carnivore.
That's totally fine. I love to hear about what people like to do outside of science. And you mentioned games specifically. This is something that I think is a ton of fun, to just find something, like you said, to turn your mind off, to think in a different way. What are some of your favorite games?
I have one in particular that I'm pretty tied to, and it's this Best Fiends game. It's on multiple devices. I happen to have an Apple device. And they're cute little characters. They're all bugs. And they're made really cute. I've actually learned a lot about my little characters and that they're actually really creatures that are in the world. But basically, you're trying to like match and make strings of little icons to beat your levels. But I've actually learned a bunch about entomology from a little game.
Oh, fun and educational. I think that's the best mix. So let's talk about your scientific side next, Samantha. You do some phenomenal work there at NIST. How do you describe what you do to someone who is not familiar with the Institute or not familiar with your particular area of research there?
So I would have to start with giving people a sense of what is NIST. I would not be shocked if almost nobody has heard of NIST and be grateful if anybody has. So NIST is part of the U.S. federal government. So we're a federal government agency. Unlike agencies like FDA or NIH or CDC, we're not part of Health and Human Services. We're part of the U.S. Department of Commerce. And the goal of NIST is to really help improve and build strength in U.S. competitiveness and technology. So one of the ways I describe this is that when you plug something into an outlet in your home, you're not worried about being electrocuted. You're confident that you're getting the right current and that it's safe. When you go put gas in your car and it's a gallon of gas, you're comfortable that you're getting what you're getting and that if you get a gallon of milk, it's actually a gallon. And when you do any number of things in life, that you're not worried about that being wrong. And why not? Why should these things be right? Whose job is that? That's kind of the purview of NIST, where are you sure that the amount of electricity coming out of that outlet's correct? Are you getting the right answer here? Are you getting the right answer there? All the things in our world in commerce. And it's not just commerce. The work that I do is in the biosciences areas. When you go to the doctor and they give you your cholesterol or they give you your glucose levels, how do you know you're getting the right answer? There are people at NIST that work on the controls and the standards to make sure that that information is correct. So that's what NIST is as an organization. My specific work is in this area called genome editing. And people may have heard of that term, maybe not, maybe heard of the term CRISPR. And these technologies, this capability is being used to try to take cells from somebody that has a disorder, particularly one that is encoded in their DNA, that's making them ill in some way, and be able to fix that thing about their DNA that's making them ill and give their cells that are now fixed back to them as a medicine. And that is incredibly mind-blowing that it could be even possible. But the question is, how do we know that we're doing only what we intended to do? How do you know that you're getting the right answer when you're checking this potential therapy is what you need it to be? And that's the kind of work that I'm doing at NIST, is to help this community know if using these very exciting technologies that can help so many people are giving them the right answers when they go to analyze these products.
So, Samantha, can you tell us a little bit more about the potential of this genome editing technology and why you feel it's so important to be investing your time in developing these resources and standards related to it?
Yeah, so genome editing is the capability to be able to have biological cells that are functional and be able to manipulate them, in particular to be able to change their DNA sequence in a way that helps make some product that's going to be really useful and have those cells still be functional. And so that's just a very, very new kind of concept. And there are lots of applications. Some of these are to make better, more robust food sources, like plants that are more drought resistant and can grow in climates, particularly as climate is changing. This can be one of those things. To make biological products like therapeutics in a more robust and efficient way. And one in particular is to be able to make human therapies to help people that have genetic disorders that previously had no option for the possibility of the word cure. A lot of these illnesses, people are treating their symptoms, trying to help them manage what's making them ill, but not ever to be able to fix the thing that is causing their illness potentially permanently. One particular example is sickle cell disease, where it's been known for decades what causes this illness, but we never had a tool that could actually help fix what's causing these people to be ill because it's an issue at the DNA sequence level. And so just recently, there is now a therapeutic that has been approved by FDA. It's also approved in Europe, and it's to be able to take blood cells from people that have this disease called sickle cell and be able to fix the DNA sequence change that's making them ill or to compensate for it and give them back their own cells as their own therapy so that these people can live normal lives and for normal durations of life where that isn't the case for the people with this illness. And so I'm super excited that this is just the beginning of the impact this kind of technology could have.
Well, I think this is wonderful. And I think very important work that, as you mentioned, perhaps goes unnoticed in everybody's daily lives. So I look forward to going into some of the details as we go through our conversation today. But I'd love to touch on motivation. I think as a scientist, there can be a lot of work to get done in a day, and it can help to have little quotes or sayings or just things that keep you going. So Samantha, do you have a favorite motivational quote or saying that really motivates or inspires you?
There's one that I think about a lot, and it's one of the models that I try to live my life by, which is it's nice to be important, but it's more important to be nice.
Oh, I like that. How does that reflect in sort of how you live your everyday life?
It's the idea of it's not just me on my journey doing whatever I need to do to get my science done. It's all of us. It's a community. We're all people together. And being able to have good human relationships and interactions, it's one of the most important things about science, because science is a community. There's no one person that has all the knowledge that they need and all of the resources that they need and all the capability to do the most important things. And so we've got to be able to work together, and we've got to be able to be kind to each other.
Well, I couldn't agree more. And I love the statement that you made that science is a community. And I think sort of leaning on each other and helping each other get through science is important. So when you look back on your own career, are there particular mentors or role models or just people who you looked up to who really helped you get to where you are today?
This might sound very stereotypical, but as I look back, I really look to my father. And I say that because my father was very visionary. And my family is actually an immigrant family. So my parents immigrated to the U.S. from Jamaica, and I happen to be born in the U.S., but I have a brother and sister that were both born in Jamaica. And I look to my father because my father gave me boldness to do things that hadn't been done before. Nobody in my family had done anything in the sciences. And so there wasn't a track record of comfort to lean on there. And the idea that you could go and you could do things and you could do really big things. And that, as my dad said, we left our island home to give you all a chance and an opportunity. So take your opportunities in this place where anything could happen and you could be anything. And so that to me was really one of the biggest drivers for me, particularly because of my background.
Oh, I think that's wonderful. And I'd love to hear what perhaps got you first interested in science. So can you take us all the way back to the beginning of your journey, Samantha, and tell us what really lit that spark for you?
So as I think back, I had always thought about being a lawyer when I was pretty young. For whatever reason, I was the talker in the family. Mm-hmm. And so I always thought about being a lawyer. And one day I remember just sitting around with my dad and flipping through the channels. This was in the 90s. And on the Discovery Channel, there was this show called The Forensics Files.
Oh, yeah.
And we stopped and we watched it. And these were real scientists, forensic scientists that were trying to solve real cases working with the police. And I remember watching it and going, oh, my goodness, this is the most amazing thing. And so we got into watching it regularly, and that really sparked my, oh, this is really interesting. Not only just forensics, but the DNA side of it. Oh, you could know information about people. So that really sparked my interest in science. And I ended up going into the field of human genetics as my core field. And that also has a little bit more of a personal origin as well, is that I'm actually bi-ethnic. So I'm both of the Black background as well as Indian, that is East Indian. And then culturally, my family's from Jamaica, so I'm also West Indian. And when I was small, people would always look at me and say, what are you? And I'd say, well, I'm a girl or I'm a person. And I didn't really understand what people were asking. And as I got older, I got the sense that I was other. And it really sparked my interest in what makes people what they are, what makes people what they look like, what people perceive of them. And that very passively, as I learned about DNA later, really connected those two things for me, where there was a personal set of experiences over my life and then this really interesting thing about science that connected me to specifically want to go into human genetics.
Well, I love this story, Samantha. Do you think your dad was surprised as he saw this interest in science emerging in you?
So yes and no. So my dad was in business. He was a CPA accountant, and he always thought, oh, you'll go that way. But he always wanted a doctor in the family. So on the other side, he was quite interested, but my family wasn't really very aware of, the nuances of Ph.D. scientist kind of what one might say doctor versus medical doctor. And so the concept of a medical doctor was more so what he was thinking. And then I explained what I wanted to do. I'm like, well, I could still be a doctor in that area. And then he's like, oh, okay.
Right. You've got the title, it counts.
Yeah.
So then can you tell us how you went from this young person getting excited about science, sort of getting interested in genetics, to where you are today? What were some of these key turning points?
So I had a very stereotypical process, and then I didn't. So in school, went through high school. I knew at that point I was really interested in sciences. I didn't know that I loved genetics yet. So took a lot of science classes. I knew that I wanted to go into biology. And so I did that at Loyola University. Previously, it was Loyola College at the time that I went there. I think it was my second year. I took my first molecular genetics course, and I was like, this makes so much sense. It doesn't to the average, logical thought process that A's, G's, C's, and T's somehow make everything about us. But it clicked to my mind, so I knew I found the right spot for me. And what happened is that after that, I knew I loved this, but the next step is usually people will go do a PhD if you know you really want to do research, which I did know I wanted to go more towards research, not towards medicine. And I just didn't feel like I knew exactly what I wanted to do yet. So I did something that is becoming more common, but wasn't as common when I did this, which is that I said, I'm going to get a job. I finished my degree. I'm going to get a job, be a technician, learn, get a real sense of what I want to do. And I just had this idea. I'm like, I'm going to go get a job, and somebody's going to support me to go do whatever additional education I want to do. Because I heard people do this in business. I heard my other colleagues saying, oh, I got a job and they helped me go get my MBA and this. I'm like, I can do that too in science. And everybody's like, that does not happen. So I was like, well, we'll see. So I went and I got a job. I applied for a job. It ended up being at NIST. I didn't really know what NIST was, like most people when I joined. Fell in love with what the mission is of NIST, because being able to help people know that the answer's right and improve their lives, even if I never get recognized, even if they don't know that I'm there, was still to me a very noble mission and an important thing. And as I was there, I realized I need to know more to have the impact that I need to have. So while I was working full time at NIST, I did my master's at night at Johns Hopkins. It was a program that allowed for you to do your master's as a scientist working. I did that. And I had a very unique opportunity. I had, who was then the boss of the about 800 of us, tap me on the shoulder and say, you should go do your PhD. Oh, wow. And I said, I like my job. No, thank you. And then I had the boss of the about 100 of us tap me on the shoulder and say, let's talk. You should go do your PhD. We see something in you, and we want to be able to support you. And I said, I love my job. I don't want to quit my job. And I was given an amazing opportunity where I was offered the opportunity to do a detail. I didn't have to quit my job. I was allowed to go do my PhD in whatever area really excited me, and then come back to NIST and help build our programs in biology. And so that's what I did. Came back, and while I was away, really got exposed to this burgeoning area of genome editing and capabilities. And I said, if this stuff really works, it could change everything about how we do biology. And I think we should make the investment of NIST helping the possibility that this technology could work. and pushed and eventually got the buy-in and be able to build and grow. And I think now being able to do some things that are impactful to help the community.
That's absolutely amazing. So what has it been like for you then, kind of going back to NIST and really building out this genome editing program? It sounds like almost from scratch.
It was exactly from scratch. I came back to NIST and I was really excited about genome editing. I'd gotten some exposure at Johns Hopkins, being able to work with and exposed to things like zinc finger proteins, which were one of the earliest iterations of genome editing technology that are still very powerful in use today. And then these things called talons, which if people know about this space, that was another thing. And then the discovery of the function of CRISPR-Cas9 and just the beginnings of this explosion of what that could mean for the biology field. And I came back and I was like, I know everybody says the thing that they're excited about will be really important, but I really believe that this is going to potentially pan out and be impactful. And so it took a bit, as people are learning, and this was very, very new. It hadn't really gotten mainstream or really outside of the geneticist world, even in the science field. And so there was a lot of building and growing. I had to meet people in the community and connect because I knew nobody and there was nobody at NIST doing this. So I really had to be that person to get into the community, meet people, socialize what NIST is, and really form an idea and try to sell people on the idea of NIST could help bring everybody together to work together on what you all need that is pre-competitive. So the things that every one of you need to know what you're doing to make your products, but it's not the thing that is your product. So it's just like for most people, they don't make microwaves, but people need microwaves to heat up their food, or people need a stove to make the food that they're making, even though, this chef or that chef might have their amazing proprietary thing they've invented. So it's the kind of idea of like, you all need a stove and you don't have a stove. So let's work together to make the stove so you all can make really great food. But in this case, it's more like medicines and products. And so both the getting internally, like this is something we should do, as well as being able to meet people and having like nobody that knew me in the field, and then selling this idea and then not having actually the program yet, but selling this idea of we could do this together. Let's all work together and make this happen was really kind of that big thing to try to get over and really introduce NIST to this community that wasn't as known in this particular community.
Certainly. Well, Samantha, I love the cooking and food analogies. I think that really helps put things into context. And I think you've had a really cool career path that not a lot of people sort of go this route to get to their ultimate position here, but you're able to do some really exciting, really important work there. And I'd love to be able to talk about some of your current projects, or perhaps if there's a particular project that you are extremely excited about at the moment. So what are you working on these days?
So I just mentioned about getting everybody excited about working together. And one of the biggest things that I can tell about is from those efforts, we now have a NIST Genome Editing Consortium. And this is a way for any kind of organization that's working on genome editing to work together with NIST, with other organizations to identify what are the key controls that you need, and us to make those controls and for us to test them and make them available then not just within the consortium, but to the broader community of people that are doing genome editing. So as of right now, that consortium has 52 active organizations that are part of it. And these are organizations within the U.S. as well as outside of the U.S. And we're working together to make physical samples that, either cells, biological cells, or DNA and vials that have key information that you can use to make sure that your test is working. Data. A lot of what's going on is, and especially I think people are more familiar with this now with this boom in AI, is that for these experiments, you have sample, you do something with your sample to identify what DNA changes you made in these cells. But all of that goes through the process of putting that data through some sort of algorithm, and the algorithm tells you what it analyzed the answer to be. And those algorithms, we need to make sure that they're working well. And so we've got a lot of effort on control data that we can test these different algorithms, making sure they're giving the right answer. Controls about how do you describe the experiment that you did? Where did you get your stuff from? What vendor did you get it from? What lot was it? All of these things that we think are probably very well defined aren't yet in this particular field. We're trying to help that because that'll help with better communication, better interpretation, more reliability. And then also the last part of what we're doing in the consortium is, it wasn't before, but now there is an international standard, globally harmonized terms and definitions for this field of genome editing. When we started, there was no textbook definition of genome editing or gene editing or anything surrounding this field. And we filled that gap so far so that people can communicate more clearly and efficiently.
And more precisely, too. I think that is critical in science. And I think we're really making the case here, Samantha, that the tools that you use and the products that you use in science matter. And we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of flow cytometry antibodies and matched isotope controls. For scientists developing biological assays, running immunophenotypic analyses, or characterizing and isolating cancer-initiating or stem cells, Innovative Research's deep catalog of flow cytometry antibodies stands ready to support your research and provide you with the accurate and precise results you need when you need them. Visit www.innov-research.com to view the entire line of Innovative Research flow cytometry products. And returning to our conversation with Samantha, I know making these new standards for technology, technical language, and also these other areas of work that you do is really important, but also really challenging. And I think oftentimes people only see the final products of science. And perhaps in your case specifically, the work that you're doing may be helping kind of behind the scenes rather than really being featured in the spotlight. So I think it's really important to highlight these challenges, these struggles, and just the process of doing science. So Samantha, can you tell us about a time that you had a major failure, or maybe you had a major challenge that you had to overcome, and how you worked through this difficult time?
Yeah, I mean, there's so many examples one can think of, and I think anybody in science can go, oh my goodness, which one do I pick? Mm-hmm. I will say one of the biggest challenges was, I think sometimes we can deal with, as scientists, the idea that an experiment didn't work and it's disappointing, or that something didn't work. But one of the biggest challenges is when the voices around you tell you it can't be done. It will never work. It doesn't. And so I had to deal with a little bit of that in this idea. It's very idealized, this idea that I could get all of these people to work together and it'll happen. And I had many people tell me that'll never work. These for-profit companies would never agree to that. There isn't a way to make that happen. And that was a struggle because I had to decide whether I believed enough that this could happen or whether I should give up on this idea and contribute in a different way. And so I think that when the worlds of discouragement and science merge, that can just compound. And I am glad that I found encouragement and belief in well, we should try. Sure, it's going to be hard, but we should try. We should figure things out. And in the end, working with my colleagues, had to pioneer a new mechanism that NIST didn't have previously in order to make this happen. I had to invent an approach that could let people come together in a consortium. And there was innovation on the document legal kind of agreement side that I never thought that I would have in my career. But that was one of the hardest things I had to deal with because I had to decide if I was going to keep trying.
Now, did you have a backup plan if this were to fail, or was it sort of all in?
I was all in on this because people work together all the time. There's in all sorts of different ways. So I'm like, to me, it was one of those, this is done. We just don't know how to do it in this circumstance. But it's not that it's something that people don't do in the world. And so I figured it might take time. There are lots of logistics. I don't know exactly how, but I think it's doable.
I love that. And I love that attitude that you went in, that it's worth a try. And I think you identified an important problem. And I love to hear that you sort of developed these innovative ways to find a solution or to come to a solution. So it sounds like this had a happy ending, but we love celebrating these success stories in science. And Do you have another story of a success, whether it was a big win like this one, or even just a small recent success that you are still celebrating?
Yeah, so I mentioned this briefly. To me, one of the biggest wins is the idea that there is now this harmonized, internationally standard document for genome editing vocabulary. And you mentioned this earlier. It takes many, many years, and it is hard to describe how challenging it is to get a set of people who are experts to agree to a standard definition, including punctuation. Mm-hmm. The amount of time I spent figuring out, is it and or is it or? Is there a comma here or not? And the amount of time we went back and forth really to make sure that it was technically accurate, sound, what we like to call future-proofed, that the things that we don't know will be invented and discovered to the best of our ability. Will this still stand and be technically accurate? And to have that be something that the international community engaged in, and we were able to, as the U.S., the consortium, and then the U.S. via me leading that effort, have this become an international standard, really, to me, is something that we've added something of value that's useful. And, I really feel that as something that's a success to celebrate.
Oh, absolutely. So how did you celebrate this success?
By telling myself I can now think about the other project.
Crossing it off the to-do list and moving on.
So we have these 3 working groups in the consortium, by motivating myself to say, and now we need something equally as valuable for the other working groups.
I think that's wonderful. So for a project like that, I think it's also difficult because language sort of evolves over time. And particularly, like you said, trying to think about these future-proof elements. How do you know when this sort of project is done? Or do you have a plan to sort of revisit in 5 years and see if revisions need to happen?
So for international standards through the International Standards Organization, you have to revisit for that exact reason that things change. We might have learned something that would change the way that words are used, or maybe you need to add to the terms and definitions. And certainly this field of genome editing is accelerating so fast. I always have to tell people now, please remember that we locked down this list of terms in 2018, and these other things weren't invented yet at that time. So they're not on the list. So certainly we will revisit that list, make sure that things don't need to be adjusted based on how words are used now or what we've learned, as well as needing to expand that there's new technology that wasn't invented then. So those are both parts of making sure that it is still relevant and usable and valuable.
Well, I think that is a very exciting success. And I know having these successes, like you alluded to, can just motivate you to kind of dig in even deeper and continue working in the lab even harder on these different projects that you have going on as well. But we try to encourage listeners to take a step back, to enjoy a moment to themselves and to give their mind a break. So we love sharing book recommendations with everyone that we have on our show. So do you have a particular book or a series that you've really enjoyed that you can recommend for everybody today?
In general, there's a lot of thought and intensity, at least in the work that I do in my science day to day. And so when I think about a book, I want to sort of turn my brain off a little bit or be engulfed in a story. And so there's a series that I have fallen in love with. It's called the Thursday Murder Club. And this is actually a series set in a retirement home where you've got people that have lived most of their life and they're now retired in this retirement home coming together with a lot of experience that they've lived. And, in each book, some sort of murder happens and they're trying to solve it. But there's a lot of humanity and human element and how do people respond to different situations in this series that to me is very gripping because it feels very much like, I have to deal with that sometimes. Sometimes it's a bit cathartic. So I really enjoy the series, and there's wonderful character development within the series.
Oh, wonderful. So the Thursday Murder Club, was it?
The Thursday Murder Club is the name of the series.
Excellent. Well, listeners, we will have to check out the Thursday Murder Club, add this to our own reading lists. We appreciate this recommendation. And I think One of the other things that we haven't yet touched on are some of the opportunities that scientists get to travel. I think this is also a big part of the career, particularly if you're working on projects with international scope like you are in your case. So do you have a place that your science has taken you, Samantha, that was the most memorable or your favorite?
So I still have travels yet to come that I haven't had yet. Mm-hmm. But of the ones that I have had, I have loved being in Paris for science. It is, to me, I mentioned earlier about how much I love cooking and eating. And so gastronomically, it's a wonderful place to be. Mm-hmm. I also am what is more than fan, but less of addict, whatever that is, of dark chocolate and chocolate in general. And so it's a wonderful place for desserts and sweets. I have a sweet tooth and just a beautiful place to be. It's also scientifically the home of a lot of deep science. And just a little plug in terms of NIST, weights and measures. We used to have the pound at NIST, like a physical artifact that weight was calibrated to. Mm-hmm. And in Paris, they have the kilogram.
Oh, wow.
And would make sure that these things are comparable to each other. We've redefined the definition of mass so that it's not tied to a physical artifact. But up until a few years ago, there was there still is, it's just not how we define weight, the kilogram in Paris and the pound at NIST. And so there's very, very rich and deep science there. And I've had the opportunity to go there for scientific work, but just a beautiful place, lots of lovely food options.
That is wonderful. Well, listeners will have to see this cultural landmark, the kilogram in Paris, that was not on my travel itinerary originally, I will admit, for the city. But what scientific occasion brought you to Paris?
There was some joint conferences that I had, a genome editing conference that I was going to have in London. And then for this consortium, I have a particular member organization that was in Paris. And we were trying to do some initial analysis of whether we would get trained to do some of the measurements that they do at their organization at NIST, or whether they were going to do them at their institute. And so I was there to learn about the technology, see how it's run, and then decide on how we would partner forward for them to contribute to the consortium. And then there have been other conferences that I haven't had the opportunity to attend, but others that are in the cell and gene therapy kind of medicine space that have been held and will be in future as well in Paris.
Very cool. Well, Samantha, thank you for sharing some of these travel adventures with us. And I think one of the other things that's wonderful about traveling is you get to meet people from all over the world. And I think this is wonderful in terms of just networking as a scientist, meeting people from all these different places with lots of different backgrounds. But I think the people you meet in science don't necessarily meet the stereotypes that you see in the media, these sort of white coat clad, very serious people who are always doing things in the lab. So we love to share the human moments in science by talking about the quirky traditions that spring up or just the fun and funny memories you share with colleagues. So do you have an example of one of these stories that you think really captures that human side of science?
One that pops to mind is when I was in grad school. And for those of you that have been in grad school, you can resonate with this. For those of you that haven't, you have those times where you just can't think about your science anymore, mm-hmm. and you need a break. You need that mental reset. And one of the things that would happen for us is that we had a certain vendor that, for whatever reason, part of their marketing is they would have little bobblehead characters, and the characters were sort of famous people in science, but they were not just bobbleheads, but they were able to stick down to surfaces. So we would every once in a while do a lab dance party. It was just one of those things where we're all like, okay, we've had enough. Mm-hmm. We're going to put on some music. We're going to have a dance party. Maybe we'll just wipe down some surfaces. But another thing that we would do is there's this thing called a belly dancer. And it's a piece of instrumentation that it's an orbital rocker. So it's flat and it basically rotates in a circle. It's a legitimate thing that we use a lot of time in science when you need to move liquid in a dish and make sure things are washed and exposed and you need movement. And so we would take these bobbleheads that had the suction cups on the bottom and we'd stick them down on the belly dancer and turn it on and let them dance. And well, we put some music on for a little bit and just all of us went, okay, let's all breathe and just have a little fun at our dance party.
Mm-hmm. Oh, I love that. I love that they were invited to the dance party too.
Exactly.
Now, did the characters have names? Did you use the actual scientist names?
We didn't give them additional names. I can't remember all of them, but there was definitely an Einstein one.
You can't not have Einstein. Oh, I think that's so cool. So are there any fun traditions at NIST or things that the group does together to sort of relax and maybe take a break from the science?
So I like to push balance. If we're not balanced people, we can't do the best. This is something that's fairly important. And so nothing uniquely in particular, but, I like to make sure that we have lab lunches, lab breaks. I like to cook. And I'll also bake things, so I'll bring things in. Let's just have some snack and not talk about science. You don't have to talk about yourself if you don't want to, because it's always everybody's purview to share as much as they want or as little as they want about things that are personal or outside of science. But to be able to share some food together and just take a break. I haven't come up with anything more fun to institute yet for our particular lab group, but at least here's something sweet, or sometimes we'll share, I had this and I loved it, and I'm bringing it in to share.
Oh, I like that. I think food is a remarkably powerful tool to bring everybody together. Well, it's wonderful to hear more about the lab environments, both where you went to graduate school as well as how life is at NIST. And I'm curious, for a lot of people who maybe haven't had the experience of working in a government-affiliated laboratory, how is it maybe different from your experiences in the academic research world at like a university?
Yeah, so the federal government has a lot of connection and impact that is a little bit different than the academic sector. And just for other people to know, there are also a lot of things that you can't do as an employee of the federal government. So on the first hand, it is the kind of thing that as the federal government or NIST, where this is our particular role, we're connected a lot with industry. And I remember in my academic lab setting and talking to others, it's more common as an academic to be connected with other academics and less common to be collaborating and working directly with a bunch of companies. And so that's a unique kind of thing that I'm experiencing, where I'm working directly with these companies, including academics, but not just academics. And then also for the federal government, there are rules that are federal government wide that, as an academic, you could get invited as a speaker to go somewhere and they give you an honorarium or they give you some funds that's yours personally to pay you or give you some resources for accepting that invitation and giving those presentations. That's not something that you can accept as a federal government employee. You can't accept funds for doing what is your job that you are being paid by taxpayer funds to do. So those are some things that you can't do. Also, another little tidbit so that people know there's so much wide ability to do a lot in science and very flexible. But one thing you cannot do that you can do as an academic is you can buy food with money that you have. As a federal government employee, we cannot use any of our funds to buy food.
Interesting.
So anytime that we have parties and things, whether it's for the holidays or something like that, the federal government is very good at potlucks.
That's fair.
Because that's the way that we can do things.
Well, it's good that you like to cook then, Samantha.
That's right.
It's wonderful to hear about some of these quirks and tidbits, because I think, not everybody gets to experience every different job. And I think giving people some insights into how these might be different can be helpful in helping people identify the best path for them. And I know in science, you are tackling some tough problems there at NIST and making some important progress. But I think even at a government institute, there are limitations, things like funding, staff, technology, time, and just the feasibility of these big problems you're trying to tackle. So if we took away the barriers, Samantha, that normally hold you back, what is the one research question or one problem that you would want to tackle first?
This is a very interesting and existential question. Mm-hmm. And, I think that what I would do is to be able to get to some answers, right? This is a hypothetical. There's no limitations. We're going to solve something around women's reproductive health. This is not my area. We just removed all of the barriers in this hypothetical. It's not my area, But I feel like it's a part of science that is extremely understudied, where there are many things that affect almost half of the world's population. And the answers that people get is, that's just the way it is. It's part of being a woman. And that there are real underlying medical needs and issues that are just understudied and not understood versus it's just the way it is and always has to be. And I really think that's an underserved thing that if I could do anything, that could be very impactful.
Well, I love this hypothetical research project, and I think you're absolutely right. You've identified a crucial area of health for, like you said, nearly half of the world's population. So a wonderful and worthy cause for this unlimited funding and resources to be applied to. So we appreciate you dreaming big with us on the show today. And we've talked about a lot of different things, but I'd love to end with advice. I think for our listeners out there who are perhaps on their own journeys, scientific or otherwise, it can be helpful to have advice to overcome some of these obstacles that may pop up in the way. So what is the best piece of advice, Samantha, that someone gave you that you can pay forward to our listeners today?
That somebody gave me would be my graduate advisor, Andy McCallion at Johns Hopkins, would say, be the best you can be. If you are in the lab, do the best that you can do. But when you're at home with your family, when you're with your friends, be there. Be the best friend, the best person at home that you can be. Don't only be a scientist thinking about your work when you're with your family, when you're with your friends. But, be the best version of you for that aspect of your life when you're in that moment.
Well, I love this advice, and I think it really resonates with our mission as people behind the science to emphasize that scientists are these multifaceted people. You're not just a scientist and only a scientist all of the time. So thank you so much for sharing that. And is there any other last piece of advice or perhaps a last note of inspiration, Samantha, that you'd like to leave our listeners with today?
So this is something that I have had to learn over time, and rejection can be hard. But one of the things that I've had to learn I can pass off to people is, when you go like, should I ask, should I not? A lot of times I'll say the worst thing that can happen is they say no. And you're exactly where you are right now. So it's okay to be bold and ask questions, because if the answer is no, then now you know, but it could be different. And so don't psych yourself out and decide, I'm not even going to ask.
Oh, I think that's also very important advice. And that way you won't sort of be left wondering, what if?
Exactly.
Well, wonderful note to end on, Samantha. If our listeners are intrigued by some of the topics we talked about today and they'd like to learn more about you and the work that you do, what is the best way for them to do so?
The simplest way is if people go to their search engine and type in NIST Genome Editing, you'll be able to get to either me or the NIST website about the programs that we have there. And there'll be also contact information there if people want to reach out more, or maybe you're an organization that is in this space and would be interested in joining some of this work. That's also very welcome.
Wonderful. Well, thank you for sharing those resources. Listeners, definitely take some time to learn more about NIST and the wonderful work that Samantha and her colleagues are doing. And Samantha, it's been a tremendous pleasure to have you on the show today. Thank you so much for your time.
I appreciate you inviting me here and this wonderful service that you do for the community, helping us all to be real people and not just the idea of somebody as a scientist.
Thank you so much. You've been a wonderful guest, and listeners, it's been phenomenal to have you here with us as well. We hope you'll join us for our next episode of People Behind the Science.